Welcome grade 10s, welcome to the new show format that we have got for you. I'm hoping that you did your revision last week and that you listened to Llewellyn and you learned all about, you should know all about the cell, you should know all about how a cell changes and everything that's involved in the cell. Because today we are going to look at division of a cell, which is called mitosis. Right, we're going to look at all the different phases and we're going to have a look at why a cell needs to divide. Now, if you have a look from last week, right, I think that Llewellyn did it with you, we looked at a concept called a cell cycle. Now, if we have a look at the board here, you're going to see a very complex diagram, but I don't want you to look at it and get too scared and think, oh, I don't know what's going on. Right, I want you to see when you looked at a cell now, remember a cell is this small little living thing. And it's almost the same as you. What must a cell do? Right? A cell is once very, very young. A cell must then mature and must develop. And when it's old enough, a cell must then divide or reproduce to make copies of itself. So if we're having a look at the diagram in front of us, you'll see the cell cycle. I don't want you to worry too much. I'm going to worry too much about all these little G1 phases and the S phase. What we're basically looking at is the following. The cell is going to grow, all right? So if I have a little cell, what is going to happen? All that I want it to do is to grow bigger and bigger. And that's exactly what's going to happen. Imagine you are a little baby, right? And here during this time, all that you do is you grow and you get ready. And what are you ready for? You want to carry on. You want to make more numbers of yourself. So you are now going to go into this process that we call mitosis or cell division. Okay. Now, when we look at cell division, we're going to see that there's a few questions that I want you to answer first. When we look at mitosis are cells that are able to divide. Now, it's not going to happen everywhere in the body, and you need to understand that cell division is not only going to happen in animals, but it also needs to occur in plants. So I want you to take your books out, right? I'm going to put 2 questions up on the screen. They're actually up on the screen now for you. And I'm going to give you 3 minutes. And what I want you to do is answer the following things. Number 1, why does a cell need to make an exact copy of itself? And number 2, I need to tell me where in the body, all right, or where in a plant am I going to find mitosis? Where am I going to see that it's going to take place? Okay, I'm giving you 3 minutes, and then I'm going to come back and see how well you guys did. Okay, guys, so how did it do? How did it go? Mitosis, I'm going to be honest with you, is not one of the most easiest sections for you to be able to grasp. So we're just going to do things, we're going to try and do things as simply as we can so that you can understand the questions that we go. Okay, now the very first question that we asked you, right, was why must we make an exact copy? Now I want to show you that is very important because when we are talking about mitosis, it's not making a cell that sort of looks like it, all right? We're talking about making like identical twin stuff here. We're talking about if I have one cell, I must make another cell that looks totally exactly. It must be exactly the same. Okay, so why would I want to do it in the body? The first one, right, is growth. Now, if you can think about yourself, I'm sure you've all got some baby pictures somewhere, right? Once upon a time, you were very, very small. Now, what happens, as I say to you when we look at the cell, what happens to us? We need to grow, we need to develop. So how was our body able to do that? We can't make one cell and just expect that cell to stretch and stretch and stretch and stretch. No, we need to take that cell and we need to, if I make more of it, then I can grow taller and taller and taller. So obviously one is for growth. The second one, I'm sure all of you as well, at some time or another, have hurt yourself, right? So you've cut yourself or you've hurt yourself or you've made bruises or something, you had an operation. What happens to those cells? We actually damage them. And I'm sure if you cut yourself, you haven't got this gaping sore, like all the skin open already. What has happened? Your skin is healed, okay? What have we done? We've made new cells, we've replaced them. Also your cells, what happens to them? They get old. And when the cells get old, we want to replace them with nice new fresh ones. So I don't want to make a kind of like the same cell. What do I want to do? If I damage my skin cell, what must I replace my skin cell with? I must replace my skin cell with another cell. There we go to the exact copy again. Now, the next one is reproduction. Now, when we talk about reproduction, you will see later in grade 11, you will look at certain kinds of organisms. If you maybe did that in grade 8 and 9, I don't think so. But sometimes you get very simple organisms that look a bacteria. And there's a little bacteria. And we don't have a mommy bacteria and a daddy bacteria, so we can't have sexual reproduction, right? So what we have is asexual reproduction. And what is asexual reproduction? We take that cell and we divide it in half, and there we go. What have we got? 2 new cells. And because we've made 2 new cells, right, one cell's making more, that is going to be reproduction. Okay, so I hope that you got most of those answers, and that you now have an understanding why do we need to make more cells and why do we need to make them the same. The next question I asked you was, where does mitosis take place? Now, there are quite a few places in the body where mitosis can take place, where we need to make new cells. But what I've done in animals, I've just highlighted about three places. The reason being is that you're actually going to look at 2 of the structures this year when you look at your syllabus. If you have a look on the board here, where we're going to find it, as you'll see here, in embryos. Now, when you are a little baby, before you are developing, all right, and I can remember listening last week, Llewellyn said to you that cells differentiate. So when you were once a little zygote, all right, you were a little round thing that looked like that. Right? Doesn't look like a baby at all. But what starts to happen? These cells that make up you start to change, start to differentiate. And what do they become? They change into your heart. They become lung tissue, right? They become your skin. So cells, what do they do? When they start to divide, they make more of it because you need to develop as an embryo. And then what do? They change. So embryonic tissue is tissue that divides and then can change into a different kind of tissue. Right, the next one, and my classes always get so grossed out when I tell them this, is that you're actually getting a new skin all of the time. Right, if you have a look at this diagram over here, you don't need to know too much about the words, but you're going to hear about something called squamous epithelium when you do the animal tissues. In your skin, right at the bottom here, you see I'm going to do the pink. You know what happens? these cells divide. And when they divide, they grow more. So what happens to the cells? They start to move up, right? So my skin gets new ones made, and then another new one gets made. And what must I do? I must make place each time for these new ones, right? And that is what I do. So, but what happens as these new ones form, all these old ones keep going to the top. And when they get to the top of the skin, look what happens. They dead. All right, they did. And then when you're in your sheets at night or in your bed at night and you rub against things, all your skin falls off, all right, and it goes into your bed. And that sometimes little hohos can come and eat it, right? So that's why you need to always make sure that you clean your sheets and you keep your windows open and those kind of things. So our skin divides, right? And what happens? The top, all the nice new cells are at the bottom. The older ones come to the top, and then they are going to divide, all right? They're going to die off. Now you're going to learn about the skeleton this year. When we look at the organ systems, you're going to look at a skeleton, and in the skeleton, you're going to look at what a bone looks like. Now you're going to learn this later on, but I just want to show you that in your bone, you have these little areas over there, right? They are called epiphyseal plates. And remember I said to you that you are going to grow. That is the area where mitosis occurs. So if we've got an area, and what happens when you grow, those cells divide, divide, divide. So what happens to the bone? Have a look over here. All right, what happens to the bone? The bone starts to get longer. So the more cells we put in, all right, what happens to our bone? It starts to increase in length. So if it starts to increase in length, what do we have? We are then going to have more. We're going to have more growth. Okay, now one of the things we always tend to forget, about plants, because I know the kids in my class, whenever I say we're talking about plants, they're like, oh, gross, ma'am, we don't like plants. But unfortunately, you guys are going to have to learn about plants this year. This term that we're going to look at now might seem very weird to you. You've never heard about it before, but when we start to learn about plant tissues, you're going to hear, all right, about this word. Now, when we are looking at a plant, this is a plant in front of us over here. A plant has got a top, right? A plant has got a bottom, and a plant has got sides. Now, when we look at a plant, what must a plant be able to do? A plant must be able to grow roots, okay? A plant must be able to grow leaves, all right, and branches. Now remember, this is not magic. A plant doesn't do chunk and then it just appears. What are we going to find inside the plant? We're going to find special cells. What are they going to do? They are going to give rise, they're going to divide, and what they're going to grow into roots. All right, or we're going to have cells that divide and make leaves and branches and stems. And what does this plant always want to do? A plant wants to grow upwards to the sun. So what do we find at the top of a plant? We are going to find cells that make more of each other so this plant can grow. Okay, I hope you have taken all of that in for the moment. We're going to have a quick little ad break, and then we're going to come back, and I'm going to have your thinking caps on because we're really going to look at the in-depth phases for mitosis. Have a quick break. I hope you had a good break and you had a bit of a walk around and you jiggled all those muscles and you cleared all the head, because now if you have a look at the board, you're going to see a whole lot of words that are totally foreign to you. They are new terms that you have to know, and as you know, biology or life science is a different language. So you guys need to be able to know these terms, right? We don't always need to explain them, but you're going to see, when we look at the phases, we need to see them in the context of the drawings. Okay, so I have got a few terms in front of me, all right, and they are, because sometimes they're very difficult to pronounce. They are karyokinesis, cytokinesis, a chromosome, a centromere, a chromatid, a centriole, and spindle fiber. Okay, what I want you to do is I want to open up your books and I want you to go and see if you can find, all right, what each of the words mean. I'm going to give you 2 minutes to quickly do it. I don't want you to write down a lot of things. I want you to quickly jot down something that you might be able to understand. Okay, 2 minutes and then we're going to mark. How did it go? Some of the terms I know are very difficult, right? But don't worry, we're going to explain them to you, and I hope that you then will be able to understand. If not, I'm sure you can ask questions and we'll be able to answer them for you later. Okay, first term that we had to do. I want to draw a little diagram for you so that you can understand when you look at a picture. I think maybe some of the terms might be easier for you to understand. Okay. When we draw a cell, I'm going to draw a very simple cell. I'm going to draw a cell and I'm going to draw a nucleus. Okay? Now what I've drawn is, I've drawn some cytoplasm and I've drawn a nucleus. Now technically, basically, all that we want to do is, we're not going to go into the in-depth things. When a cell divides, what do I need to do? I need to now make another nucleus. Okay, so what do? I must take that nucleus and I must divide it. But when I divide it, I need to make sure that I keep the same number of chromosomes. Okay, I also need to take the cytoplasm and I need to divide the cytoplasm in two. So if I were to draw that, then I would have one nucleus, and then I would have half the cytoplasm and half the cytoplasm. Okay, so when I make a copy of all my material inside my nucleus, I call that karyokinesis. What is it? Division of the nucleus. Karyo means nucleus, and kinesis means to divide or to split. Okay? So karyo means to divide my nucleus. Cytokinesis is to divide my cytoplasm. Cytoplasm, kinesis. All right, so there's my cell. So what do I need to do first? I need to, all right, divide the nucleus and then I divide the cytoplasm. Now you heard me talking about a chromosome, and you've heard a chromosome, but you have no idea what it is. Okay. In your cell nucleus, you have this large structure called the DNA. Now, a DNA is this long molecule, okay? Now, when a cell starts to divide, only when a cell starts to divide, this long piece starts to unravel. And when it unravels, it looks like a chromosome. So it's like it loses its water, and what it does, it shrinks, and it looks like a chromosome. Here you see we've got, how do we make a chromosome? There's my chromosome over there, okay? Now, in actual fact, my chromosome should look like that, one whole little thing like that. But we're going to see in mitosis, what we're going to have is a phase called interphase. And during that phase, now you guys need to think about it. If I have got sweets, I have got 4 sweets, and I want to give 4 sweets to you, and I want to give 4 sweets to me. How am I going to do that if I've only got 4 sweets? You know what I do is, I make a copy of those 4 sweets. Okay, so in my nucleus, you're going to learn about it more when you go to grade 12. In your nucleus, that DNA, it makes a copy of itself. So instead of having 4 sweets, how many sweets have I got now? I've actually got 8, okay? Because what have I done? I made a copy of it. So have a look at this chromosome. If I make a copy of it, there we go, I now have 2. But you know what? It's very difficult to try and keep track of things if they are all over the show. So what we do, we take the chromosomes and we bind them together by what we call a centromere. Okay, so for the centromere. So that centromere, all right, is then going to hold the 2 pieces together. Now those 2 pieces, that is 1 chromosome over here. But what is 1 chromosome made of? 2 identical pieces called chromatids. Okay, called chromatids. And that is what a chromatid is. Here's 1 DNA, here's my 1 sweet, there's my other identical sweet. And what I want to do is I want to keep them together. So I take an elastic band and I wound them together and I keep them together. So although I have 2 sweets, with that elastic band tying them together, I actually have 1 that looks like 1. Okay, and a centriole, if you can remember when you did the animal cell, what do we find? We find little centrioles, all right, they're little structures in the cell, and what they are going to do is they're going to produce spindle fiber, okay? And spindle fiber is like little, imagine a spider and its web. How does a spider catch a fly? It spins out this web, all right, and then when a fly gets stuck into it, then it pulls it in, all right, and that is how we're going to move the chromosomes. Okay, so I hope you sort of understand, but when we start doing the phases, it's going to be clearer to you. Now I want to start with the phases. We're going to do 1 or 2 phases, then we're going to take a break, and then we're going to carry on with the last of the phases. Now I try to, if you can, I try to remember a bit of a rhyme. Okay, so if we have a look at mitosis, what you're going to notice is the following. It actually happens very smoothly, but when it does happen, we can see that certain things happen at certain times. So we put them all together, we're going to get the whole process of mitosis. Okay, so I want you to remember IPMAT, right? I-P-M-A-T. Now, I stands for interphase. Then I'm going to go to a prophase, then to metaphase, then to anaphase, and then to telophase. Now, you have no idea what they are, but all I'm going to show you now is we're going to look at this whole process. Now, what you must keep in mind, I want to take my nucleus and I want to make 2. Okay, listen carefully. I need to take my nucleus and I need to make 2 copies of it. All right, so we're going to start off with it. I'm going to show you a little table. Now, you need to be able to do 2 things. You need to be able to look at a diagram and you must tell me what is happening there, because we see at a certain phase or stage, certain things must happen. Okay, so let's start. I for interphase. Now here is a diagram of a cell. Now when you see this phase, you will see it looks like any cell. Why? Because this is the cell. We go back to the cell cycle. This is when the cell is growing and it is getting ready to divide. Now, when it's before it divides, you will never, ever, ever see chromosomes. What is it? It's still in the nucleus and the DNA is still twined as my chromatin. So because I can still see a nucleus, because I see chromatin, I can then say my cell is still getting ready. It's not ready to divide yet. But what it does do, which we cannot see, very important, it makes a copy of the DNA. Makes a copy of the DNA. The DNA replicates. Okay. Now we go on to the next phase. Now imagine it's slowly, it's smoothly, it's starting to happen. Those of you who've got access to YouTube, go onto YouTube, go and look at mitosis. You will see a lot of wonderful videos, right, showing you how mitosis occurs. There's a lovely group called Amoeba Sisters that I really like, where they tend to show the process in quite a very simple way. Okay, now have a look at the next drawing. Now if you see this drawing, prophase, all right, remember what were we looking at? Mat. Have a look at this diagram. What are we starting to see here? Oh, look what I see for the first time. I start to see chromosomes. Okay, so now this is really my first true phase of mitosis. I have these chromosomes, and now you just need to understand that the chromosomes, they now become center stage. They center stage of my cell division. And what do? Have a look here. Can you see this dotted line over here? My nucleus disappears, or my nuclear membrane. Everything in the cell disappears, because the only thing that we want to see is we want to see the chromosomes. Okay, so the chromosomes, they are there. The DNA is unwound, because now these are my sweets. Right now I can start to see my sweets. Here I've got my 4 sweets. They've all got the elastic bands around them, and I'm ready here, and I have my sweets. I'm ready to divide it between me, and I'm ready to divide it, my sweets, with you. What do I need? All right, I need my centrioles to move to the poles. If my cell is round, I want to separate it. And how do we separate things? Top and bottom. And if you look at our Earth, what do we call? North Pole, South Pole. We call extremes, all right, one ends of each other, we talk about the poles. And so the centrioles start to move to the poles, right, because ultimately what they're going to do, remember, they're my spider web. What are they going to do? They're going to pull it up, right? They're going to attach to the chromosomes and they're going to pull it up. So if you have a look, how do I know it's prophase? Look here. Look at my key points, and then we're going to have a break. I can see for the first time, I can see chromosomes. That is important. Where we go, I can see chromosomes in prophase. What makes it different from all the other phases is that my spindle fiber hasn't started to form yet. It's just the centrioles are moving to the poles. Okay? And I also know that it's prophase because what do I need to do? To be able to do everything, I need to clear everything out. So that nuclear membrane needs to go. So the only thing that the cell can concentrate on is these chromosomes that need to be, all right, separated. We're going to have a quick little break, and then when you come back, we're going to have a look at the last couple of phases, right, of mitosis. So go and have a stretch again, get all that blood working and the brain cells firing. We'll see you shortly. Welcome back. I hope you had a nice little break and you got all the blood going again. All right, we're carrying on now from looking at our phases of mitosis. And now we said, all right, remember what we're looking to do. I've got my sweets, and here are my sweets, right, in the elastic band. And what do I ultimately want to do? I want to give you 4 sweets, and I want to keep 4 sweets. All right, that's what we're looking at, because that's what I want to do. I want the same number, and you want the same number of sweets. Okay, so now what we did was we noticed that the cell, it doesn't just automatically divide. It has to get ready to divide. And we quickly looked at the phase of interphase, where the cell, we can't see all the things going on. It looks like a normal resting cell, but the cell is getting ready to divide. It's making copies of that DNA, which are the chromosomes, okay, and it's getting ready to say to you, here guys, here's your 4 sweets, and yay, I get my 4 sweets. All right, so what do we need to do? We need to actually, it's so much easier to split things if we can put them into separate bodies. So this long DNA unwinds, and we see that it looks like the chromosomes. Now, if you have a look at the next phase, remember Ip-Mat, interphase, prophase, metaphase. Now, if you have a look here, you'll see I've highlighted M, and metaphase starts with M. Have a look at where the chromosomes are in the cell. They all line up to the middle. Okay, now there's a reason for that. Because remember, what do I want to do? I want to now, right, give half to one side and the equal half to the other. Now imagine you cutting something. If I want to give half to you and half to me, do I just take something and cut it? No, I don't. What do? I line it up nice and neatly, all right? I make sure that everything is in line, and then I cut down the way, which makes things equal so that I can have equal parts. So imagine looking here, why do we line? Okay, because remember, my sweeties are bound with an elastic band, and they're lying here. So imagine what I do is, all the sweets are lying there, and I take one knife, okay, and I go, and I cut the elastic bands. What have I done? I've now freed the sweets, because what are the sweets then able to do? The sweets can then move to you, and mine can move to me. So there's a reason, right? What we need to do is, we all jumbled up. We now need to meet at the middle, right? Because once we are in the middle, what happens? I break those elastic bands. I break those centromeres. They're broken, and look what happens, okay? Your sweets start to move to you, and my sweets start to move to me. Okay, why? Because what is happening, right? Remember that spindle fiber, that spider web that's attached to the chromosomes? What are these guys doing? These are like fishermen, okay? And what are they doing? They're pulling, they're pulling, they're pulling. And when they're pulling, I'm pulling my sweets to me, which are the chromosomes. And what are you guys doing? You're pulling on your centrioles, and you are pulling your sweets, which are the chromosomes, to you, right? Because remember, what did we want to do? We want to take one, and we wanted to divide it. And that's exactly what we've done. And we call that phase anaphase. So Ip, interphase, prophase, metaphase, anaphase. Okay, anaphase. So now we need to round things up. We need to end things. So we've got interphase, prophase, metaphase, anaphase. And our final phase, where our division of our nucleus, our karyokinesis, all right, is done, is the telophase. Now look what you can see happening here. Have you got your sweets? Yes. Have I got my sweets? Yes, thank you. We finished. The only thing I need to do, remember our sweets were the nucleus. So once I've got my sweets, I don't need to, all right, I don't need to divide anymore. So what happens? My nuclear membrane starts to reform, all right, and what do you think happens to these chromosomes? They're going to jumble up to chromatin network. What happens to my centrioles? They do not need to be needed anymore because the spindle fiber has done its job. It's pulled it to the opposite poles, right? So what do we need to do? I now need to divide my cytoplasm. And when I divide my cytoplasm, right, that is cytokinesis. Now look how many cells I started off with. All right, if I go back to the top, I started off with 1 cell. At the end of mitosis, I started off, or ended off, should I say, with 2 cells. Now, what do you notice about the cells? They are identical. They look exactly the same. So, how many sweets do I have? I have 4. How many sweets do you have? You have 4. All right, so have we got 2 identical sets of sweets? Yes, we do. All right, we've got 2 identical sets of sweets. Okay, now we're going to look at a few examples of questions that you might be able to get in one of your tests. Now, if we look at the first one, you need to understand, guys, you need to be able to look at a picture and see where exactly does it fit in, where along the phases, and why can I recognize it as that phase. Okay, so let us start with the first question. It says, study the diagram below, this is this diagram over here, showing a specific phase. It's not telling you, right, it says to you, you need to know what this phase is. All right, and undergoes during mitosis, and answer the questions that follow. Right, so now look at my cell, and I see, and automatically, right, what do I need to look at? I see that the chromosomes, wow, where are they? In the middle. Right, they are in the middle. Right, so what does that mean? That if we answer our first question, what phase of mitosis is shown in the diagram above? M stands for middle, 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 middle. Right, and remember, let's go back. I'm going to go to the previous slide. What do you notice? What's the phase where they're all in the middle? Yes, well done, those of you who were able to get it. All right, it is called Metaphase. All right, what I want to do is I'm going to give you 1 minute, one minute, okay? A quick 1 minute. I want you to answer question 1.2 and 1.3 for me. I want you to label A, B, C, and D, and I want you to tell me how many chromosomes are in the cell. I'm giving you 1 minute. How did it go? Were you able to answer it? Remember I said to you in the beginning, there's a little bit of terminology that's going to come in here, and you're not going to be asked to expect to understand all the terminology. But you will see here, you're going to be able to label the diagrams, and that's where the terminology is going to come. Let's see how you did. All right. Number A, I hope you guys all got cell membrane. Now, I wonder if lots of you said nuclear membrane. I hope you didn't. Why? Because what happens during prophase? The nuclear membrane disappears. Okay, so what we're looking at here, we're looking at the cell membrane. Number B, they're showing you the whole structure of that, and I hope you labeled it a chromosome. Number C, have a look here, it's at the poles, and what do we see coming from number C? Spindle fiber. So number C, oh, there we go, is centrioles. All of a sudden it's gone to black. Oh, I think I'm pressing buttons here. Number D, all right, is the pink. Look at it, that is your spindle fiber. All right, I hope you guys got that. So you must be able to label your cells. Always a good thing, right? Those are always nice, easy marks if you sometimes don't understand the concepts. The last question was, how many chromosomes are in the cell? Guys, you have a look. You count 1, 2, 3, 4. There we go. There are 4 chromosomes inside of the cell. Okay, so there we go. We've gone through that one. Now listen carefully. All right, I'm going to give you 1 minute again. How many chromosomes will be found in each of the daughter cells? Read carefully. How many chromosomes do human body cells have? And then what they want you to do is draw a fully labeled diagram of part number B. I'm only giving you 1 minute. All right, I want you to quickly, because the first 2 will be quick, and then I want you to quickly draw a rough diagram of what question number 1.6 is. How did it go? I hope you can remember. All right, some of the things that we looked at the beginning, even one of the slides would have showed you what you actually had to draw. Okay, so let's have a look. All right, I asked you how many chromosomes. Now you mustn't get confused. How many chromosomes are there in here? There are 4. Now the word here that was the trigger is a daughter cell. When I have a cell at the end of mitosis, I have 2 cells, right? And I call them daughter cells. Why daughter? Right? Because a daughter is not yet mature, all right? It is, we say it's a younger version. So in other words, how many chromosomes will be found in each of the daughter cells? In other words, how many sweets did you have? I had 4 sweets. How many did you have? And the answer would then be 4. Okay, the answer would be 4. Because what is the mitosis? If I start off with 4 chromosomes, each of my cells must have the same number. Okay, next question. How many chromosomes do human body cells have? And human body cells, right, we have 46 chromosomes, or we have 23 pairs. Okay, 23 pairs. Now the last one is also again, all right, a nice easy one. If you can remember in the beginning, I showed you a diagram, all right, of a chromosome, but they asked you here to draw a fully labeled diagram. So you can't just draw a chromosome, you need to do labels as well. So I hope yours looks something like this, okay? Doesn't matter how you draw it, Mine's going to look something like that, okay? And I'm going to label it. This is a chromatid. Right? And its exact copy is on the other side. Remember, what was a chromatid? An exact copy of each other. Oh, my nose is itchy and I need to sneeze. Sorry, guys. The next is what holds it together? It was my elastic band, was my centromere. And what is this whole thing there? That is my chromosome. Now it's a diagram. They might not be too finicky, but I think when you do a diagram, what I want you to do is you put a heading, or you can put it underneath, okay? A diagram of a chromosome, because you've told us that you know what part B is, and you should have known it from doing your labels as well. So a diagram of a chromosome. Here you've got a chromatid, a centromere, and you've got a chromosome, and you have drawn it correctly. Okay, our time is almost up. I want to go into the next question, because here you're probably going to go, oh, all right, why? Because this looks a lot more difficult. Okay, so you need to have some simple diagrams, but I'm just showing you below that what we can actually do is we can actually take proper diagrams of a cell dividing, and we can ask you questions on that. All right, so if you have a look at the different stages, all right, what should you see? I'm just going to show you. Look at the nuclear membrane. It's still there. That would be prophase. Look at the chromosomes on the equator, the middle. That would be metaphase. Look what's happening here. It's a plant cell. So a cell wall is dividing and they finish dividing. That is telophase. Or I would say, you could also say cytokinesis. And have a look what's happening here. They're being pulled to the poles, and that shows you anaphase. All right, grade 10s, thank you for today's lesson. I'm afraid you need to go and practice. Go and look on the YouTube. Go and find some videos on mitosis, because this is quite a difficult section. Until next time, keep well.